but now next up is Dr. Vicente Yepes, and he's a researcher and scientific manager of the chair of computational molecular medicine at the Technical University of Munich. And he's really a leading world expert in multi-omics approaches in both diagnostics and research of rare diseases. And he has also been very active in many European initiatives and projects. So please, the floor is yours.
Great. Thanks a lot for the introduction, and Alex for the invitation. I guess he's here. I cannot see him. And well, it's an honor to share this session with Kim, and you will see later that I mean it. So I do have some conflicts of interest. I'm co-owner, co-founder, and full-time employee of Omics Discoveries. So you will know the journey of a patient that has rare diseases, right? Since the first symptoms until genetic testing, it's a lot, until diagnosis, if there is, and in many cases there's not, right? So our job here is to, or my talk here will be how we reduce the time for this and to increase the patients that actually receive these genetic diagnoses. So for that, well, there were many different technologies that we'll talk about, and the idea is that these tools, these technologies reach the whole world, not just high-income countries. So genetic variant interpretation remains limited, right? So I can call variants with the best technologies out there, and I can see all the rare variants of the genome. But that doesn't mean I know what's the effect of them. So for that, a useful complement is to perform multi-omics and actually be able to measure the effect of each of these variants and not just predict it. So there's various levels of evidence for diagnosis, DNA, so to actually find the variants that are consistent with the mechanism of the disease. Then, of course, the clinical phenotype. So the disease-causal gene or genes have to match the clinical presentation of the patient. Correct segregation, so it has to match the mode of inheritance from father, mother, de novo, recessive dominant, and so on. And if multi-omics experiments are performed, it also has to match what was found and confirm or complement the genetic initially finding. So how does this work, more or less? You can come from the DNA, and there you can perform exome sequencing, short-read genome sequencing, now long-read genome sequencing, or optical genome mapping to try to find the variants of interest. Then from the omics side, you can do RNA-seq, methylation, proteomics, metabolomics, lipidomics, and for sure there will be much more in the upcoming future. And you always take care, in the middle there's the phenotype, right? It always has to match the phenotype. And then you can go from one side to the other, so what I would call the DNA first. Given that we found candidate variants, we evaluate in the omics. So we do this, what I call this focus testing, that we go to the gene of interest where this variant is and try to test it with another assay. Or we can go the other way around. If no candidate variant was found, then we do this multi-omics testing, this discovery mode, and then go back and perform. So for example, if only exome sequencing was performed, then we can perform genome sequencing or long read or OGM or relaxed cutoffs, filters, and so on until we find the variant that was suggested by the omics experiments. And there's various ways of doing this. So one is sequential, that first you did the DNA, you found nothing, and then you do the multi-omics. That's maybe the most cost efficient, but it's also the one that takes more time, right? And you have to wait a couple of weeks until you failed, recontact the family, maybe they come, maybe not, do again a blood test, for example, and do another assay and conclude. The other one is in parallel, which is quite convenient. But the best one, and the one that we should aim for, is to have it integrated. So to analyze the DNA, the RNA, the proteomics in the context of each other and use the information together to better and more confidently reach the diagnosis. So I started defining shortly the difference between short read and long read, and here's when I said that I meant that it's an honor to share this session with Kim. Also, most of these slides were taken for this amazing review that she wrote last year. And for everybody that is starting to do multi-omics, I very much recommend to read this one. Like it's very comprehensive. It's great. So you see here the genome sequencing, and you see the short read, the long read. I guess you all know what it's about. And you can very easily see why the long read it's better for example, better mapping or differentiating between genes and pseudogenes, for repeat expansions, for structural variants, and very importantly for phasing, right? To be able to confirm that if I have two variants, they're indeed in the opposite alleles. Optical genome mapping, maybe less of you are familiar with it. So it's another technology that is not really sequencing, and it provides a bigger overview of the genome, so bigger chunks. So it's very well suited for deletions from around 500 base pairs to the full chromosome. And the idea is also to assemble these fragments into the reference, and it's quite good at detecting large deletions, inversions, translocations, basically large structural variants. So I'll start with a bit of comparison, so you know that whole exome sequencing is relatively good to detect the short variants and mitochondrial DNA. CMVs, it can, but it struggles a bit. With short-read genome sequencing, you can now detect the CMVs more properly, but certain structural variants and short tandem repeats is not the best, and mosaicism. With long-read, basically you can do almost everything, including this phasing that we saw, and optical genome mapping better suited for these large variants, but not for the short variants. So you will not be able to find an SNV or an indel with optical genome mapping. So that's for the DNA. Now we go for the multi-omics. So RNA-seq has been widely used for rare disease diagnostics. And the idea is that here you start by sequencing RNA from a clinically accessible tissue. So it can be blood. Blood is the most widely used. But in many cases, it's better to use skin-derived fibroblasts because more genes are expressed there, or muscle for patients that have muscular disorders. Then the idea is to detect this aberrant expression, aberrant splicing that can take multiple forms, such as the ones we see here. The first one is exon skipping case, the second one an exon truncation, the third one an exon elongation, the fourth one an exon creation, or monoallelic expression, which consists on measuring, given the two alleles, how much of each is expressed. Finally, we integrate with DNA because RNA-seq alone does not enable diagnosis, but you always have to integrate back to DNA. And this has increased diagnostic rates for around 5 to 15% in many studies throughout many centers, many diseases, many tissues, thus confirming the validity of this complementary technology to DNA. And here the idea, and this applies for RNA, but also to any other omics, we are not very much interested in comparing two groups, right? Disease A versus disease B, or unaffected versus disease, or parents versus affected kids. But the idea is to model a same population and then find this outlier. So it's a bit similar to DNA, right? That we are interested in very rare variants. So also in omics, we are interested in very rare events. So we model the population to find really the outlier that can explain this, the disease. And for this, like for DNA, in which we have this reference database of millions of patients or healthy individuals, also a sufficiently large cohort is needed to properly model and get the background distribution for the different omics. For proteomics, a bit similar. So proteomics is not sequencing. This one, for example, is mass spectrometry based. I took this figure from a mitochondrial disease review. So that's why it has this mitochondrial protein of interest, but it can be any protein of interest. And what's cool about proteomics is that it does not allow only to measure the protein itself, but also complexes. For example, this C1 is a complex 1 of the respiratory complex 1 inside the mitochondrial subunits, and even many, right, for these oxals, mitoribosome, whatever, and then it allows even a nicer or a broader confirmation of the effect of the mechanism of the variants and not just pinpointing it. This figure is from this review from Charlotte, but there's many other papers confirming this and showcasing the added value with respect to DNA and also with respect to RNA sequencing. There's many more, like here, metabolomics and lipidomics. The idea is similar to extract, to quantify, to normalize. I didn't mention that before, but there's usually lots of batch effects that can be, or these covariation effects that can be both technical and biological. Technical batch, for example, lab, sequencing kit, machine, and so on. And biological, including ancestry, sex, tissue, biopsy origin, age, so on. And again, after normalization, you quantify, and the idea is to also try to find these extreme values that, for example, not found in the parents if it's a de novo case. again, taken from this awesome multi-omics review. So coming back to this figure, we saw that the DNA will, if we analyze multi-omics data, we will get VCF files from DNA, from short-read, long-read, etc. BAMLETs, we'll get mitochondrial DNA variants with heteroplasmy, CMVs, each with different visualization, structural variants, translocations, inversions, tandem repeats, phasing, and multiple scores, the CAT score, the Revel score, genome allele frequencies, by population, and so on. There's many things to know that I'm sure most of you are familiar with. With the phenotype as well, there's many ways of associating, quantifying it, for example, using HPO terms, imaging, and so on. With segregation, you will now write a pedigree, de novo, consanguinity, many things to consider. And in multi-omics, there's also many things, many scores that you can get, fold change intensities, p-values, set scores, and each of these with their own plots, and each of these with their way of integrating with DNA. So you see, it's a lot of data, a lot of results, a lot of information, a lot of files, plots, and it's difficult to be an expert in all of them. So let's say we are all collaborating, and you send your data to the data analyst, and you can receive this back, the results attached to an email, the tables in Excel and the plots, and it can come with a nice description of what it is, but still it will be extremely difficult if you're not experienced with that to conclude. So what we did in Solvathons is, instead of this attaching tables to emails, is to organize these solvathons. So what is a solvathon? It's a structured workshop. That's the shortest definition. And it's basically a hackathon, a coding hackathon, but not to code, but to solve cases. We did 3 days, both on-site and online events. Very importantly, it has to be multidisciplinary so that everybody can attend. So analysts, interpreters, geneticists, bioinformaticians, clinicians, PhDs, postdocs, everybody is invited and welcome. And it has, so the first goal was, of course, to accelerate the diagnosing of samples, but it was immediately clear that there was this second goal that came with it, that was this knowledge transfer from all directions. That was very important so that people can go back and keep on analyzing and now become experts on all the technologies. Other features from the solvathons is that they offer a focused environment, allowing this rapid interpretation, and very importantly as well, this mutual motivation. To be in groups helps a lot reaching the goal. And very importantly as well, it helps building trust and collaborations. Many of the people I met, I still work with. And of course, why not have fun? So it's not easy to do one of these events. So there's 3 phases to it. So before, the idea is that the data is collected, sequenced, the results are prepared. And it's very importantly is to have this data analysis freeze. So what does that mean? There should be a way of selecting which patients are going to be part of a solvathon. For example, 20 families will be, these 20 families will participate, and we will do short-read exome with proteomics and RNA-seq, and that's it. Like we have to adhere to it so that it actually happens on time. And then the analysts start preparing the results with the help of the interpreters so that we are sure that it's on time and we don't delay or lose time during the event. Now, during the event, the clinicians, they present the results, the clinical side of the cases, so that we all in the room, we know what we're dealing with, which kind of diseases. They're usually quite complex because they meant they were not diagnosed locally. So it's quite interesting because you learn a lot on both directions. Then the analysts, they explain what they have done, which tools have they used, which results have they produced, and very importantly, they make this use case. So it's basically one slide with how a diagnosed patient looks like. I'm going to show some of those later. And the idea is that then the people follow these instructions, and when they start looking at it, they also assemble this slide according to these guidelines and try in that way to prove the diagnosis. Then we form small groups, 5 to 10 people, and then we start this interpretation, and it's important that these groups are also heterogeneous, combined of different kinds of participants to also share this knowledge. After the event continues, after the 3 days, it's very important to continue the interpretation. So to organize, of course, this cannot be on site anymore, but online meetings regularly, maybe every week, every 2 weeks, to continue and try to analyze all the patients, if not all families were concluded during the event, to try to at least reach a conclusion for all of them, which can be a diagnosed candidate or unsolved if nothing was found. And the idea is to also have these results interpretation freeze. I'm going to talk about that later. So this is what we tried to do in Solvathons. It was definitely not easy. So there's this data freeze that I said, well, all the cases that must be prepared before the Solvathon. Then during the Solvathon, we are in groups, we solve some cases, some we mark as candidates, some we just couldn't find anything, we discard. And after, maybe we said that a date of 2 months after the Solvathon, where we said we should really check all the cases, go through everything, also to justify the funding, right? There's usually a large funding before that, and they want to know what happened, like really with numbers, not just, oh yeah, it was cool, right? And this allows you to say, these many cases were inspected, these were analyzed, but we continued afterwards, and we reached a conclusion for all the participating families. And it's important that people that attend these events, they commit to this from both sides, right? From the analyst side, that are going to work extremely hard, especially the last few days before the event, to have everything ready and safe, all the data and results ready to be shared just on time for the event to start. And then the interpreters, that they are really going to check every single of the findings and give feedback back to them. So they say, yeah, this was useful, this was not really useful, this was redundant, so that we can improve and in the next events be better and provide better information, better results, more tailored, more useful for the people that are actually analyzing and interpreting it. So we did 4 Solvathons performed during Solvathons. The first one was focused on RNA sequencing at the CENAG in Barcelona in Spain. It has around 60 participants, half of them online and half on site. The second one was about short-read genomes. That was the biggest by far in terms of participants and data, so that was performed only on site. The third one was about long-read genomes and optical genome mapping in the Radboud in Nijmegen, Netherlands. And the last one, multi-omics, we went back to the CENAG in Barcelona for the last event of Solvathons. A bit of numbers, so again, around 30 participants on each of the on-site ones, and way more in the online one, the second one, about short read. Hundreds of families, and this is quite important. So it was not expected that we go through the 300, 200 cases, but it was expected that at least everybody from each of these ERNs, the European Reference Networks, that are the ones listed here, these for epilepsy, neuromuscular disorders, cancer, Ithaca, immunological disorders, and neurodevelopmental disorders, that each person at least attends so that they can go back and resolve their cases. And yeah, we saw that there's lots of participation from the different ERNs part of Solvathons, and it led to a total, I think it was 28 cases solved on site. It was extremely difficult to really force, in this case, or the clinicians to give feedback afterwards, so it's hard to estimate exactly how many were solved in the next weeks and months, but we believe it's at least 70. So now I'm going to really go case by case and explain what worked. So for example, for the RNA-seq in the first Solvathon, we were there on site. There was a patient, a neuromuscular patient, in which one base pair deletion, so a frameshift, was identified in this gene nebulin, inherited from the father. This is a recessive gene, so the second variant was missing. Then RNA-seq was performed and releasing the results of the Solvathon, and it was found this 80% expression reduction. And this very high reduction is indicative of a biallelic loss of function variants. So we knew one from the framesheet, but we could not find the other one after having performed whole exome sequencing. Then RNA-seq revealed a created exon, and this was not present in any other sample of the cohort. The second exon contained the second variant that was, of course, missed by whole exome sequencing, and it was later proved that it was inherited from the mother, thus reaching the corresponding diagnosis. So why was a Solvathon needed for this case? Is like, if we just provide this plot, and this plot is not so easy for a person that is seeing this for the first time to reach all of this conclusion and diagnose the case. Another example, this involving repeat expansion. So when we went to the first Solvathon in February 2023, I had no idea what were these. So I was in charge of the RNA-seq results, and then what we found is that there was, in one gene, this NOP56, there were 2 patients that had very low reduction of this. And in these 2 patients, before they were done the standard DNA analysis, and no SNVs or indels were found. But the clinician who was there on site, she immediately recognized this gene and said, wait, in my lab, we just published a paper that associated this NOP56 with this kind of a spinocerebellar ataxia. And the variants that caused it were these short tandem repeats. So then on site, we ran this tool expansion hunter, where we indeed found these repeats, and we got 2 diagnosed families in less than 2 hours because we were on site. Otherwise, there's no way through an exchange of emails we would have reached this conclusion, or maybe we would have, but after at least a month. So a great example of why being on site helps, and we all learned how to analyze, how to conclude. So next time, maybe that we see this, we will be able by ourselves to conclude, or to at least know where to look for. The third case, there was this large 9 megabase per deletion found in short-read genome sequencing data in chromosome 2 of an epilepsy patient. Inside this region, there is this gene, NR4A2, which is dominant, and it's known to cause epilepsy. So it was quite clear that this was a very serious candidate. So now my question to you is, like, if you see this in short-read genome sequencing data, would you trust it? Do you diagnose as is, or do you do any other experiments? So if not 100% of you answer yes, then it's good to do some confirmation. In this case, RNA-seq was performed. And you see here the set score among the whole transcriptome, and there's something clearly going on chromosome 2, that when you zoom in, you see that all genes inside that region are aberrantly expressed, are done underregulated by around 50%, which is expected, given that there was deletion in just one of the 2 alleles. Another case, quite interesting, and this is RNA, mostly RNA-seq, but it can be any other omics. It can be proteomics, metabolomics, whatever, that something like this extreme was found. So there was a patient suffering from a neuromuscular disorder, in which by whole exome sequencing, no candidate variants was found. So we performed RNA-seq, and again, I emphasize that this can be any other omics, and we saw a very low extreme reduction in the gene DMD. For the people that work here with muscular disorders, you know that if you find this in a patient with neuromuscular disorder, you know that that has to be the disease causal one. That is, there's no way around it, even if variants were not found. So whole exome sequencing was performed initially, then we performed short-read genome sequencing, and a translocation was found between DMD, which is in chromosome X, and BCAS3, which is in chromosome 17. This was not possibly to be found by whole exome sequencing, and RNA-seq, in this case, guided the finding via whole genome sequencing. For the next example, I'm going to shortly introduce to this circus plot from BioNano from optical genome mapping that I guess not all you are familiar. So it's quite a cool visualization. It contains like all the chromosomes, and on each of these bands, it showcases some kind of structural variants that can be this SVs, copy number, this absence of heterogosity, variant allele fraction, translocations, fusions. This is a fusion, quite interesting, that goes from one chromosome to another, or it can be also inside the same chromosome. And then what you expect to find in this plot is that it's empty. That means there's nothing. If there's whatever going on, dots, lines, arcs, it means there is something. Just a short introduction. So there was this one patient with a quite complex syndrome, 4-year male proband, mostly with severe global developmental delay, hypotonia, vision problems, quite more. The parents were unaffected, so we suspect either compound heterozygous or de novo. And whole exome sequencing and short-read whole genome sequencing was performed with inconclusive results. So as you expected, I was going to show this circus plot. And then many things were found, more than 20 de novo events, including here in copy number variants in chromosome 12, in 9, 16, and translocations going from within chromosome 1 and also, chromosome 12, and from chromosome 12 to chromosome 3, which is a lot. So It's really a lot. It's not common to see this amount. So as a person who, like, you send your data for optical genome mapping, you have never worked with that technology, you receive this plot attached to an email, you have no idea what does it mean. Is that good, bad, makes sense, it's quality control issue? No idea. So now that the optical genome mapping expert was there, she said, wait, yeah, this sounds relevant. We are able to zoom into chromosome 12, so we not just see the whole, but like a better view of just chromosome 12. And indeed, we see many copy number variants and these translocations within, including many genes that seem to are plausible that all of them, or in combination, or each of them, it's hard to say all out of this how the mechanism of action of the disease is, but it seems that this indeed is the disease causal. And you could ask, so why was this not found in exome or genome sequencing? Actually, these were found at certain levels, but because so many were found, these were actually discarded as a quality control. So it's quite an interesting case. We were discussing yesterday evening with the corresponding clinician, Beatriz, and Fulia, an expert who know way more than I about this, and then the conclusion was that it's one of these two, these large chromosomal rearrangements involving replications, deletions. Not sure which of these two is, but probably is one of those two. You can read this review that we found yesterday that can explain this disease. So I hoped with this case I convinced you how interesting this is, and there were many lessons and challenges. So first of all, if you're going to organize one, you have to mind this data privacy and consent, right? There's many people that are going to see the data, so be sure that you have the right consent for that. Fully online events are cheaper and can involve more people. However, it's better to do it on site. So if you can, just do it on site. And then it's very important to have this consistent structured documentation from the interpreters for both diagnosed and also discarded cases, because then, like, you know it's discarded and you don't check it again, lose your time, spend hours, and then, yeah, somebody else had discarded it or even diagnosed. So the best is that every single patient that gets analyzed gets properly documented following a common framework and not just free text. Then I mentioned already about this data freeze must be finalized, agreed well in advance so that we work together towards it. And after the event, it's very important that there are these consultation hours guided by a results interpretation freeze to keep the momentum and keep diagnosing cases. Future directions, but this is not the end of the talk, there's more coming. The idea is to keep on, right? These events were quite successful inside Solvathons. Now there's Erdera, Jardin, the national programs. But you don't have to think big at the continental level, but you can do it just the regional or even at the hospital level. Organize a 2-day event where you gather everybody in the human genetics department to solve cases. And yeah, of course, the standardization of tools, interpretation frameworks. It will be great that these events become a hub for showcasing and using and showing how to use the state-of-the-art technologies and tools. And partnerships with healthcare systems and funding bodies, because these events are expensive, so they need funding. And develop collaboration with undiagnosed hackathons, which is the second part of the talk. Yeah, these were the 4 different ones we got. You see, we're quite happy due to the success of the events. So for the very complex cases now, you can attend these undiagnosed hackathons. I had a video, but I'm going to skip it due to time. So in a nutshell, what are these undiagnosed hackathons? It's very similar in many senses to the Solvathons. It's an intensive on-site event, solving the UDNI and the Undiagnosed Disease Network International from the most challenging, so selected cases that were very difficult to solve, and they were actually not solved in the respective countries. Of course, the idea is yet to solve these cases and find collaborations across all over the world. And I mean it all over the world. There were clinicians from Pakistan, Congo, South Africa, Ecuador, Mexico. Like, it was amazing. And lots of experts, more than 100 from all over the world, and again, very multidisciplinary clinicians. And this was great because there were patients. It's the first time in my life I actually saw patients and families, I as a bioinformatician, right? So it was amazing to see them on site. Again, before the event, the participants are selected, and Angelica here worked very hard to have the data ready for the first day. And then also so that once the event starts, we gather up in teams and analyze and work with people we had never worked before, very motivated to solve the cases. Here are the two events. The first one was in Sweden, the Karolinska Institute, the second one also in Radboud in Nijmegen, Netherlands. You see here in the front row the patients, the families, out of which some were diagnosed. We'll see how many. The first one of these events in numbers, there were 10 families, more than 100 participants from 28 countries. And there was short-read data for DNA whole genome and for RNA, short-read and long-read for both of them. At the end of the event, after collaborating in groups, we reached the diagnosis of 4 out of 10 families. If you want to know more, there's the paper, or talk to Angelica and Fulia that are here. I'll show one of those cases, quite interesting one, a 6-year-old male suffering from seizures, in which there was a splice region variant found, or spliced variant found, which is relatively easy and straightforward. And the second one that we needed this genome sequencing was to find this other, the deletion. Together, compound heterozygous, and we were able to confirm the deletion with this exon skipping from short-read RNA-seq data. That's really confirming that the deletion indeed is present. This unpublished, do not disclose, please. The second one, held in Nijmegen, was even bigger. And this slide is quite cool because it shows not just the number of cases, families, but also the number of data sets that were generated. This was everything, exome, short-read genome, optical genome mapping, long-read genomes, transcriptome, so almost all the different omics that I showed today. And very importantly, a lot of work was done to make this happen from all sides, including the organizing committee. And very cool, so there were many families that came from all over the world in whatever creative way they could, even by flying the patient, taking the sample to conferences, like for example, last year to the ESAG, the event was right after, so it's, yeah, we were creative or they were creative to make this happen on time. At the end, there were 122 participants from 28 countries, which contributed altogether to 10 new diagnoses and 9 candidates. I liked a lot that there was the definition of what is a new diagnosis, because many people say, wait, in 2 days, can you really be sure about it? So it was defined this as, would you share the molecular finding with the family? And if the doctors were confident, then it will count as a new diagnosis. I'll show a case from Georgia from this patient called Anastasia, who had severe intellectual disability, dysphoria, among others. It's a great example of a diagnostic odyssey. So every possible DNA testing was performed to her, since karyotype, gene panel, whole exome sequencing trio, methylation, whole genome sequencing solo. Then yet in another company, also chromosomal microarray analysis, and you can see everywhere negative, 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 negative. Usual 6 years odyssey. Until this patient came, and the clinician came from Georgia, Tika herself. And now that you're experts in the circus plot, can you tell where the variant is? Or what's not seen? So exactly. I saw somebody saying chromosome 2, there it is, right? Optical genome mapping suggests a de novo event on chromosome 2. So this is great, the optical genome mapping, right? It immediately we diagnosed this after 6 years. Then long-read genome sequencing was also performed, and that is able to confirm also the breakpoints of the deletion in this gene SEP2, which is known to cause a Movat-Wilson syndrome, which matches the clinical presentation of the patient. Yes, so it's clear, right? It's there, it was seen by optical genome mapping. I think nobody can doubt that 2 different technologies found this. It has to be present. That's bringing closure for this patient and the family, the doctor, for 5-6 years. And then this yearly event, there's also one this year, this time in the US. You can still apply. I think the deadline is among these days. And you can apply from both sides, right? As a data analyst, so you come with your expertise in analyzing DNA, RNA, proteomics data, or by submitting cases from your local center. There's the website on. Feel free to go for it. So my take-home is super simple. Just one is this state-of-the-art technologies and tools, multi-omics, and collaboration across all types of expertise is crucial to resolve complex cases. That's my words, but you can also listen to Gareth saying that.
The Undiagnosed Disease Hackathon is global kindness in action. Experts from around the world gathered to help some of the most vulnerable children on our planet. Together, generating the new solutions, the new partnerships to help so many. Answers that will bring so many children out of the dark clouds of uncertainty.
That was the last slide. I can thank my colleagues at the Technical University of Munich, where I work, also from Solvathons, Hon, Anna, Alex, Christian, Steve, German, Lysenka. I'm sure I'm missing more people. And then from the Ninos Hackathon, of course, Mika and Helen, Angelica, Fulia, Wendy, Orion, and I guess I'm missing more. Sorry for that. And all the patients and the families, and you for waking up early on the last day to be here. Thanks a lot.